Okay, today we're going to talk about the Hardy-Weinberg principle. So there's two biology terms you need to know before you can start to understand the principle. The first is population, which is the number of individuals of the same species in the same habitat, ecosystem or area. And you've got allele frequency, so that's the relative frequency of an allele at a particular locus in a population. And this is expressed as a fraction or as a percentage. Usually in questions you have to express it as a fraction, but if it does ask for it as a percentage, then you must make sure you do that. So what is the Hardy-Weinberg principle? So it's a model that predicts two things: the frequency of alleles of a particular gene, and it also predicts that this will stay constant from one generation to the next. So in reality, there are lots of assumptions which might not actually apply, which will make the Hardy-Weinberg sort of frequencies that you work out perhaps not strictly true. So the conditions under which the principle applies, so the assumptions you have to make are: there's no mutation, there's no selection pressures, you've got a large population, the population is genetically isolated, mating is random, and there's no migration. So all of those things will ensure that the allele frequency will stay constant from one generation to the next. And as you know, in reality, these things do happen. So there are two really important equations that you need to know, and you can, or you usually have to, use both equations in an answer. So first of all, you've got P plus Q equals 1, where P is the frequency of dominant alleles, and Q is the frequency of recessive alleles. So imagine you've got a homologous pair of chromosomes here, and you've got a gene locus A at this position on this chromosome. This chromosome's got a dominant, and this chromosome's got the recessive allele at that locus. So this would be the frequency of allele A's versus the frequency of allele B's. So allele little a's. So P, big A would represent P, little a would represent Q in this equation. There is another equation, and it's important to note that this looks at genotypes. So if you think diploid, so each of these, so most organisms that we look at in questions are diploid. So if you look at the equations, you've got P squared, so you've got two lots of the allele P. You've got Q squared, so you've got two lots of the allele Q. And you've got 2PQ, so that's representing heterozygotes. So P squared, frequency of homozygous dominance, Q squared, frequency of homozygous recessives, and 2PQ, frequency of heterozygotes. So this could be represented here again. So frequency of big A's, homozygous dominance. Remember, P from before was the frequency of allele, the dominant allele. You've got 2PQ, frequency of your heterozygotes, and you've got Q squared, frequency of your homozygous recessives. We'll look at some questions in a minute, but it's important to note that you usually start off a question by trying to work out what the frequency of your homozygous recessives are. You can't necessarily go straight into working out your homozygous dominance, because, for example, in this situation with the pea plants, you've got purple flowers, but purple could be caused by heterozygotes, or it could be caused by homozygous dominance. So often you have to start off with a question of working out what Q squared is from your homozygous recessives. So here we've got an illustration showing sort of population, homozygous dominance, heterozygotes, and homozygous recessives, and how what allele frequency means. So we've got a population that contains half dark and half light. The dark is caused by the dominant allele, big A. And you can see this is made up of homozygous dominance and heterozygotes. And we've got the light, which is made up of homozygous recessives. So now genotype frequencies. The frequencies of Q squared would be little a. So there are 10 of them out of 20. So that would be 0.5. So half of those individuals would be Q squared. For the homozygous dominance, we've got 2 of them out of a population of 20, so that'd be P squared, so that'd be 0.1. And the 2PQ, the frequency of the heterozygotes, would be 8, out of 20 would be 0.4. You can also see the frequency of the individual alleles within the population. So if you counted up the total, because each organism is diploid, because there are 20 individuals, there are 40 alleles in total. So you've got 2 alleles for each characteristic. One for your mother, one for your father, in this case. So we've got 40 alleles in total, 28 of them are recessive, so that would be Q. So we've got the 20 here from these 10 individuals, plus we've got 8 little a's there. So that makes 28. So the frequency of allele little a, or frequency of Q, would be 0.7. And we've got 12 big A's. We've got 4 from the homozygous dominants, and the rest, the other 8, are from there. So we've got 0.3. You can always error check, because if you add these up, they should come to 1. If you add these up, they should come to 1. So I'd like you to pause the slide. I'd like to read through the questions. I'd like to have a go at them. And then when you're finished, unpause and we'll go through the answers. So these questions are more straightforward than you usually get in a question, but just to sort of ease us into it. So we've got population of plants with a mixture of red and white flowers. We've got dominant R for red and little r for white. We said in this question that dominant allele R has a frequency of 0.6. Well, that is an allele frequency, so that's a P or a Q, and this is dominant, so it's P. So P is equal to 0.6. We know this equation. Rearrange the equation, that must mean that Q is equal to 0.4. And now it says, calculate the frequency of homozygous dominant, recessives and heterozygote individuals in the population. So now we're not asking for allele frequencies, we're asking for genotype frequencies. So we have to use the other equation. So we've already worked out what P is, it was 0.6, so P squared is just square P, so that's 0.36. We want to work out the homozygous recessives, so that's Q squared. We know what Q was from before, so we just square it, 0.4, square 0.4 is 0.16. So that's the frequency of homozygous recessives. And heterozygous individuals is 2pq, so it's 2 times by p, which was 0.6, times by 0.4, which was q, and gives us an answer of 0.48. And then, using these numbers, using these frequencies, if the total population of the plants was 2,000, how many would be white? Well, white must be homozygous recessive, because you must have 2 copies of that little r. So that's q squared. So 2,000 times by what q squared was, 0.16, times by the frequency. So there'd be 320 white plants. Let's look at this question then. So we've got, can be used to work out the frequency of a recessive allele in the population. Hemochromatosis is a condition caused by a recessive allele. In one country, 1 in every 400 people were found to have hemochromatosis. Describe how to use the Weinberg equation to calculate the frequency of people who are healthy but carriers or heterozygotes. So if you could pause the video, have a go at the question, and when you're ready to go through it, press play. So first of all, let's see what we've got. 1 in every 400 people have got hemochromatosis. So people who've got hemochromatosis must be homozygous recessive, because it says it's caused by a recessive allele. So they must have 2 copies of the recessive allele. So they must be, this is a genotype, so that if they've got the condition, the homozygous recessive. So 1 in 400 people must be q squared. So let's put that into a decimal. 1 in 400 is 0.0025. We can then use this to find q. So you simply square root it. So square root of q squared gives us 0.05, so q is 0.05. We now find p. So p is equal to 1 minus 0.05. So remember, that's the first equation that we went through. So p is 0.95. The frequency of the dominant allele is 0.95. And now to work out the frequency of heterozygotes, which is 2pq, it's 2 times by p, which was 0.95, times by q, which was 0.05, to give an answer of 0.095. More often a question will be something like this. So 50 out of 100 have a condition caused by a dominant allele. Find the heterozygote frequency. So if you could have a go at this, please. So pause the video, I'll go through it in a second. So for this, you can't use 50 out of 100 to get P. That's a common mistake that people might make. So the dominant condition can be caused by homozygous dominance and heterozygotes. So even though it's told you so many people have this condition, it's caused by a dominant allele, you then know that the rest don't have that condition. And so the rest must be homozygous recessive. So 50 out of 100 have the condition, 50 out of 100 don't. So 50 out of 100 must be homozygous recessive. So that's Q squared. So Q squared is 0.5. First thing you usually have to do. Then you square root it. So give us 0.71. Now you can work out P. 1 minus 0.71 gives us 0.29 for P. And now we can find the frequency of our heterozygotes. 2 times by P times by Q to give a frequency of 0.41. So please remember that framework. You usually have to find the frequency of homozygous recessives first, and it usually won't tell you the number in the question, it's what's left over. So find Q squared, then square root it, now you can find P, and now you can find your frequency of heterozygotes. Okay, so if you have a look at this question, and then pause the video and we'll go through it after. So you can see here, it doesn't actually matter whether the KDR minus allele was caused by a dominant or recessive allele for the question, because it's asking for the heterozygous individuals at the end. So this is the percentage of the allele. So this has to be P or Q because it gives us a frequency of an allele. So it has to be P or Q. So 2003, 20% were KDR minus. So 20% is 0.2. So you just convert that to the decimal. So that let's say we're calling that Q, so P must be, rearrange the equation. So remember P plus Q equals 1. So 1 minus 0.2 is 0.8. And then 2PQ is the heterozygote. So 2 times by P times by Q gives us 0.32. Okay, could you read and have a go at this question? This is supposed to be a little p, by the way, so the recessive allele. So pause the video. Okay, now some people might fall for a trap, so I've got the beware sign. This does not say use the Weinberg equation. This says calculate the actual frequencies. So you have to be really careful to only use Weinberg when it tells you to use Weinberg. So in this example we've got 500 individuals. As they're diploid, so that means they've got 2 copies of each chromosome, so that means they've got 2 copies of each allele. There are 1,000 alleles for flower colour. There are 280 little p alleles from the heterozygotes, so 280 heterozygotes, so that means we've got 280 little p's. There are 400 little p's from the homozygous recessives, so we had 200 homozygous recessives, but they've each got 2 copies of little p. That means we've got 400 copies of little p there. So we've got a total here of 680 little p alleles out of 1,000, so our actual frequency of little p is 0.68. So this is not using the Hardy-Weinberg equation.